Morning, everybody. This is our Week 7 Wednesday Embryology lecture for winter 2025. Sorry I can't be there in person, not feeling the greatest. So let's get this done. This will be the cutoff for the final, or not for the final, but cutoff for test number 2, which will be next week. And it's Hummingbird. One of my favorite shots of bald eagle coming in. Those are the Calero Reservoir. That's the dad. And at your stage of bird identification, you should know what this is. And don't say a red-tailed hawk, because where's the black wing bars? Remember, red-tailed hawks have a black wing bar here. So the other choice would be a Cooper's hawk. Doesn't have orange here. That would be a red-shouldered hawk. So Cooper's hawk. That is a ruby-crowned kinglet. And where have we been? So we talked about nodal cord last time, and we really got into the neural crest cells. Talked a lot about what they become. Just a cool picture I found showing that the neural tube has formed. The nodal cord is still down at the bottom. So now we have ectoderm. The primitive streak is gone, has degenerated at this point. We're in week 4, folding has occurred. And you can see neural crest cells, and they're actually starting to wander off to do their jobs, which is mainly to make nerve and ganglia, most of the parts of the nerve. And yeah, we talked about what they give rise to. This list is not complete, but just some of the things that popped into my head. So neurons, ganglia. Remember we said that the outer coverings of a lot of nerves are not made by neural crest cells, but the vast majority of nerves are made by neural crest cells, including the axons, the working parts of the nerve. So ganglia of the PNS and ANS, pigmented cells, melanocytes. Remember, embryologists aren't the greatest anatomists or histologists, so they tend to call melanocytes pigmented cells. The parts of the skull, frontal bones, parietal bones, mandible and maxilla. Let's see, ethmoid, nasal bone, I could have added on there. All the auditory ossicles. And we said the leptomeninges. So the dura mater is not derived from neural crest cell, but most of the pia and arachnoid in the brain is derived from, or the leptomeninges are derived from neural crest cells. So now let's back up again. This is probably the 4th time we've backed up. And let me start my emergency camera here, by the way, just to make sure. All right, just in case this recording doesn't work, I have emergency camera backup, and we just kind of went through, well, we went through this stuff. I won't go through that again. We went through where have we been. We've covered all this stuff before, so this is where we are now, Mesoderm differentiation. So let's back up. Remember at the beginning of week 3, the mesoderm was formed by modified epiblast cells kind of streaking or moving through the primitive streak, getting their orders and going to different places. And they pile up in the middle. The ones that were exposed to low levels or medium levels of BMP. Was it BMP? I think it was BMP. Different BMPs. Anyway, they become mesoderm cells. And that's where we left it. So we left it at this. So this is undifferentiated mesoderm, still mesenchymal. It hasn't morphed back into epithelial cells like the endoderm, and the ectoderm is epithelial cells, but this is still undifferentiated. So after it's formed, almost immediately it starts splitting or segregating into 3 distinct layers. Okay, so this is what we're going to make next. So during week 3, the neural tube is just kind of starting to form here. We can see that neural tube would be right here. Nodal cord would be here. It's just starting to form that flying V. Primitive streak is still here. But we start differentiating pretty quickly, and they form 3 subregions. Okay, I think everything we said already. Yep, we said this already. Mesoderm does not convert into epithelial cells. They stay mesenchymal. So they went through what kind of transformation to form mesoderm cells? Just simple epithelial to mesodermal differentiation. Or that should say mesenchymal differentiation. Epithelial to mesenchymal differentiation. So fix that on number 10. All right, so if I had to, this is just a way to remember the 3 regions that form, and they'll be on each side of the midline. We have a lip is the mnemonic, if you will. It's lateral plate mesoderm, we're going from outside to in, intermediate mesoderm, and paraxial mesoderm. Very, very busy, the paraxial and lateral plate mesoderm, especially paraxial mesoderm. That's where somites come from. So we got a lot to talk about with these guys. Intermediate mesoderm, we'll see that develops the urogenital system. But that perhaps isn't as important as paraxial mesoderm is to us, because the spine comes from this type of mesoderm. So anyway, now we can fill in the blanks here. So there's our nodal cord, there's our neural plates forming, and now we have segregation. It's bilateral, and it's symmetrical. So the tissue, the mesoderm tissue closest to the midline, this would be the midline or the middle. On an axial view here, we're looking kind of straight on at the bilaminar disc or trilaminar disc at this point. So we have paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. Are we good with that? Great. And just a timeline, there's a bunch of other events going on at the same time. So gastrulation has been completed. Remember, gastrulation was building the primitive streak and the trilaminar disc. Primitive streak, in fact, is shrinking and the nodal cord is growing. We talked about that. The neuralization is just beginning at this point. So this is about the time where this mesoderm segregation starts. What's the cause of this differentiation? We're not going to go crazy into the weeds. We'll just cut it down to one transcription and growth factor type biologic or biochemical, and that's called the BMP. And we've talked about BMP4. It's a very, very busy molecule. It's like nodal. So there's a lot of stuff that needs BMP turned on and turned off. But here's the story, and we're explaining why this happened. And by the way, this diagram, there wouldn't be these huge spaces in here. The author is just trying to set things apart. There would be no space here. This tissue would be all the way up against the endoderm and ectoderm here. But why does this happen? That's what we're going to explain. Why does this differentiate? And it turns out it's because of BMP inhibitors or BMP4 inhibitors. You can just call them BMP inhibitors. That stands for bone morphogenetic protein inhibitors. And it turns out that the nodal cord and the primitive streak have their BMP4 genes turned on, and they're secreting BMP4 gene product right here in the center, and it's dripping down into the cells of the mesoderm. It's like leaving the cells and soaking into this tissue, and it spreads out. So as the BMP4 that's being created here, as it spreads out this way, it gets weaker and weaker and weaker. Of course, right next to the primitive streak, primitive node, and the nodal cord, of course it's going to be very strong in these tissues right next to the midline here, right next to the nodal cord and primitive streak. So this tissue right next to the midline, which is where the primitive streak and nodal cord are, they got exposed to very high levels of BMP4 inhibitors. And therefore, their BMP4 gene is not completely turned off, but it's almost completely turned off. But the farther you get away from the midline, this kind of dilutes. So right in the middle here, where the intermediate mesoderm forms, that got exposed to kind of moderate amounts of BMP4 inhibitors. And as you go way out here to the side, I mean, the BMP4 inhibitors get soaked up by all these cells in between, so not a lot of BMP4 inhibitors make it out here. So therefore, there's nothing to inhibit these BMP4 genes to be turned on. So the cells of the lateral plate mesoderm will have very high levels of BMP4 because they're exposed to very low levels of BMP inhibitor. And the cells of the intermediate mesoderm, they'll secrete moderate amounts of BMP4 because they experience moderate amounts of BMP4 inhibitor. See how that works? So got ahead of my slides, but let's go through it again for those of you who didn't catch it that first time around. So mesoderm regions of the new mesoderm cells get exposed to different amounts of BMP4 inhibitors as they leak out of the nodal cord and the primitive streak, and primitive nodes included there. Remember the BMP4 inhibitors, or BMP inhibitors, noggin, cordon, cerebrus 1. I've talked about them before. Came from primitive streak and nodal cord. And this exposure turns on the mesoderm cell's BMP4 gene. Well, the exposure doesn't turn on. That's another mistake. This is a new slide. It turns it off, but it doesn't turn it off completely. So we've got to fix that on slide 16. So exposure really turns off the mesoderm's BMP4 gene, but it doesn't turn it off completely. And therefore, it can express some BMP4. And the key is, the more BMP inhibitor that was soaked into these cells, the less BMP4 will be secreted. Okay, so different regions of mesoderm secrete different amounts of BMP4. It's all dependent on how much BMP inhibitor soaked in. So I'll make that correction and re-upload this PowerPoint. Or you could also say there's a BMP4 gradient running through the mesoderm bilaterally medial to lateral. Different amounts of BMP4 expression cause the segregation within the mesoderm. Great. And here's another look, kind of a head-on look of the BMP4 gradient. So paraxial mesoderm cells, para means next to, axial, and this is the axial plane, like the middle of something. And paraxial cells, paraxial mesoderm cells, they were exposed to high levels of BMP and 4 inhibitors, so therefore they don't secrete much BMP. Don't have that in that slide, but this is just looking at the BMP4 inhibitors from another look. Okay, therefore, these cells secrete very low levels of BMP4. So that's the story with the paraxial mesoderm. If you just memorize stuff, you memorize that the paraxial mesoderm was exposed to high levels of BMP4 inhibitors, and therefore they express low levels of BMP4. That's the bottom line. Okay, move out to the lateral plate mesoderm. Remember these BMP4 inhibitors, which are coming out of the nodal cord in this picture, they couldn't make it all the way out here. They got sucked up by all these cells. So lateral plate mesoderm cells are exposed to very low levels of BMP4 inhibitors, and therefore they express very high levels of BMP4. Some of the genes are turned off, so it's not full power, but it's close to full power. Creation of BMP4 and release of BMP4. Okay, and then the intermediate mesoderm, well, that got exposed to an intermediate amount, or kind of a modest amount, of BMP4 inhibitors, and therefore it expresses a modest or intermediate or medium level of BMP4. Get that? Hopefully that sunk in. Here's another picture. This time I'm just showing the cell expression, the BMP4 cell expression. So decreased BMP4. How come? Because they got exposed to crazy high levels of BMP4 inhibitor. Intermediate mesoderm, so mild intermediate levels of BMP4. And then high levels of BMP4 here, because there wasn't a lot of BMP4 inhibitors that made it out this far. You good with that? I think we can all grasp that. Okay, let's look a little bit closer now at mesoderm. Specifically, let's start with the paraxial mesoderm. So if you had to pin me down and say, give me one thing that paraxial mesoderm forms, because we're going to get specific on this, but in general, paraxial mesoderm is our chiropractic realm. It's the axial skeleton. So paraxial mesoderm, remember before we just said mesoderm, oh, it forms bone, and it forms muscle and tendons and ligaments, but now we're getting more specific. Now we know exactly where the axial skeleton came from. That's paraxial mesoderm. And there's a few exceptions, right? We know the skull, some of the, like the parietal bones and the frontal bones, that came from the neural crest cells. But in general, like most of the spine, all of the vertebrae, the ribs, comes from paraxial mesoderm. And we'll get even more specific. We'll split up this paraxial mesoderm into somites, and we'll split the somites into different sections. Also gives rise to a lot of muscle, tendon, and bone. Well, I don't need to put bone in there because we already talked about bone. So we'll be getting much more specific on this. So just another picture of paraxial mesoderm. Great. Does it form everywhere in the trilaminar disc? So does this differentiation or the segregation, does this occur like into the plane of the page and out of the plane of the page? Does it occur all the way down the primitive streak and nodal cord, or the axial pole that would stick through the center here? So it is found everywhere. So paraxial mesoderm, and it has to, because it makes vertebrae, it has to be everywhere because we have vertebrae from C1 all the way down to the coccyx. So everywhere in embryological speak is the cranial region, occipital region, cervical region, and then the trunk. And why embryologists don't get more specific. They say cervical, but they don't say thoracic, lumbar, sacral, coccygeal. They call all that stuff the trunk. And I know, but that's just, they're not the greatest with anatomy. That's what they do. Yeah, once it's formed, it's very busy. It doesn't stay in this paraxial mesoderm form. It differentiates very quickly. And specifically, it converts back to epithelium. And so this tissue, this paraxial mesoderm, went through epithelial to mesenchymal, back to epithelial conversion. So that should say, it shouldn't say mesothelial. That's my dictation software doing it. We're never talk about mesothelium here. That's always mesenchymal if you see those mistakes. Okay, that's my voice recognition, so I need to fix that one too. All right, so basically we're going to take that paraxial mesoderm, and here's an overhead look at it. And we can see that the neural tube is on its way to being formed. We have these openings, so it's not a mature nodal cord or mature neural tube yet. But primitive streak is gone at this point. But what happens right next to the midline is we start getting a clumping, an epithelial clumping of the tissue on each side. And those are called somites. In the next lecture, we'll talk a lot about them, because the somites will split into different regions. And I can actually tell you where the vertebral body comes from versus the spinous process versus transverse processes. We can get real specific about this. So these are the somites, and they form quickly, and they form all the way down from head to toe. And they go through the process of, they were once epithelial cells, they converted to mesenchymal cells, And now they've converted back into epithelial cells. And that process of making these clumped epithelial blocks of tissue, somites, it's called somatogenesis. Somatogenesis. And yep, we'll be coming back to that. What about the intermediate mesoderm? We're not going to talk too much about that because for us it forms the urogenital system. And it's important to know, but we're not going to go into the weeds on that. We just don't have time. But there's the intermediate mesoderm right there. Exposed to medium levels of BMP4 inhibitor. Therefore, it expresses just medium amounts of BMP. And AKA is intermediate mesenchyme, sometimes it's called by Moores, which is a chiropractic board book. European medical book is called nephrotome. They definitely wouldn't use that on boards, even if Larson was a board book for us, because it tells you nephro, you think kidney, it tells you, gives away the answer. So they're going to use intermediate mesoderm, or at maybe rarely intermediate mesenchyme. So it forms on each side of the midline, we know that, just lateral to the, make sure you know locations, just lateral to paraxial mesoderm. It could be a distractor. Which one of the following statements is false with regard to intermediate mesoderm? It forms medial to paraxial mesoderm. No, there's nothing medial. There's paraxial mesoderm. This is lateral going that way. Oops. So it can form medial. I mean, medial is in the middle, so that would be a silly answer choice. So make sure you know that stuff. This one, unlike paraxial mesoderm, which formed everywhere, this doesn't form everywhere. So it only forms in the cervical occipital region. I could have added in there occipital region, cranial, occipital, cervical. Oh wait, no, that's correct. Sorry, I'm trying to outthink myself here. This one forms in the trunk, so that's thoracic, lumbar, sacrum, coccyx regions. Then it forms the next one up, which is cervical, and that's it. It does not form in the occipital region or the cranial region. So nothing above cervical. Okay, so it just doesn't form there. Why? Because we couldn't possibly need it there. There's nothing related to the urogenital system. It also becomes segmented, but the segments are very, very weak, and they're not defined enough that you could see them with a light microscope. So it segments, but you have to have an electron microscope to see that clumping, and that only happens in cervical and the upper thoracic region. But it's not enough to call them somites. So there are no somites in the intermediate mesoderm, even though technically it does clump a little bit. Not enough to call it somites, or even somatomeres, which we haven't talked about yet. Okay, in the trunk region, so thoracic, lumbar, sacral, lower thoracic, lumbar, sacral regions, it does not segment at all. And that's really where something called the nephrogenic cord comes from, which is kind of the grandfather of all the, like the kidney, the ureters, the bladder, and all that stuff. We're not going to get crazy deep into that. In general, what does it form? Intermediate, definitely on the test right here. So in general, it gives rise to the urogenital system, which includes the kidneys, the ureter, the bladder, if you're female, ovaries, if you're male, testes, the urethra, so all the plumbing, as well as certain parts of the adrenal gland, which we won't get any more specific. Well, we know it doesn't form the medulla, so it's really cortex. So I guess we already know this, because we know that the medulla is formed by neural crest cells. Lateral plate mesoderm is pretty busy. So this one forms, and we're probably in about starting week 4 at this point. There's some AKAs, so lateral mesoderm, the plate is left off. Lateral intraembryonic mesoderm, that's Moore's, that's a chiropractic board book, so you got to be on the lookout for these things. Remember, we have a extraembryonic mesoderm. This is intraembryonic mesoderm. And this will form lateral to the intermediate mesoderm on both sides. So this is farthest away from the midline, or the nodal cord, neural tube, all those midline structures. It's farthest away. This one doesn't segment at all. You can look at it with electron microscope and you can't see any type of segmentation at all. And that segmentation probably has to do with how much BMP4 is expressed. So the segmentation probably occurs because of lack of BMP4 expression. So because this one doesn't or expresses tons of BMP4, then it hinders the clumping, the somite forming. So no somites of any kind, lateral plate mesoderm. Here it is, way out here. Notice the location, so it's lateral to intermediate mesoderm. It would be lateral to paraxial mesoderm. It's not medial to any structure. So if there was some other weird tissue out here, then you could say it's medial to it, but remember, medial is going that way. All right, we can also classify or subdivide lateral plate mesoderm into a cranial region and a caudal region. So the dividing point is the fifth somite where this occurs. And there's no physical line or structure that you can see dividing them, but physiologically and functionally speaking and developmentally speaking, different things happen with the cranial portion of lateral plate mesoderm compared to the caudal portion. All right, lateral plate mesoderm will also split, and this is the big one. It splits into something called somatic lateral plate mesoderm and splanchnic lateral plate mesoderm. In general, what does it form? So if you had to pin me down, what does it form? So heart. Not all of the heart, but a good chunk of the heart. Smooth muscle. Big one here. The limbs, the arms and legs of the skeleton. So that's important. Big one, gut tube. So that's your entire alimentary canal is formed from lateral plate mesoderm. And we can get even more specific when we split it, which we'll be doing here in a bit. Body wall. And we'll be getting specific on this soon. Key identifying factor of these lateral plate mesoderm cells, if you look at them and test for BMP4 expression, very high levels of BMP4 expression. So let's get a little more specific. The cranial, or the more toward the head part of lateral plate mesoderm, that's the one that contributes to the heart. That also contributes to some of the dorsolateral neck muscles, splenus capitis, semispinalis capitis, all that. Remember those things? And the caudal division is more for the bones, ligaments, and tendons of the upper and lower extremities. That has limb forming potential. So make sure you know the cranial division of lateral plate mesoderm has nothing to do with forming the lower extremities. Okay, both of the regions will form one structure. The gut tube and the body wall comes from both divisions. But the heart, dorsolateral neck muscles, cranial division, and the limbs come from the caudal division. All right, let's develop the lateral plate mesoderm. Let's split that up a little bit more and work on that now. So soon after its development, lateral plate mesoderm develops little bubbles, little cystic cavities within its substance. Very similar story to that of the creation of the chorionic cavity in the extraembryonic mesoderm. Remember, it was a solid mass of tissue, and these little bubbles, and they popped up, and then they coalesced together, and they formed a big old cavity called the chorionic cavity, extraembryonic coelams, aka. And yeah, so these little bubbles are called coelomic spaces. Okay, coelom like Tom, coelomic spaces. And there's a cartoon for visual learners we could say, oh, the tissue's got little bubbles popping up, coelomic spaces. And you can guess the story, can't you? Those coelomic spaces are going to kind of come together, morph together, coming together. Like, do we know who those guys are? Are you guys too young to remember the Beatles? Those are the Beatles back in the day. They still have good music. I think my grandkids even like some of their stuff. Anyway, so the new spaces that's starting to form, it's called the intraembryonic coelom or embryonic body cavity. There's Langman making up words again. And the folding, a week 4 story is folding of our flat trilaminar disc. That's starting to happen right when these intraembryonic coeloms pop up. And so that's just kind of a time landmark there. But now all our little cavities have kind of pooled together into one big cavity. And now we can call that the intraembryonic coelom. Okay, so now what's going to happen? Well, we've got a big old chorionic cavity out here. Right, this is forming placenta out here. So why not have a communication between these two? And that's exactly what happens. In the next stage, we basically destroy this tissue right here, and the chorionic cavity and the intraembryonic coelom will become one unit. And that's basically what we're doing. So intraembryonic coelom rips through the lateral portion of the lateral plate mesoderm, and it allows for temporary communication. Doesn't stay there forever, but we got a temporary communication between the chorionic cavity, aka extraembryonic coelom, and our new intraembryonic coelom. Cool. So we've also created 2 new layers. If I rip this through, this one's probably better to look at. If I rip this through, then we've created a new layer here and a new layer here, and it matches these layers. Remember what these are? Secondary yolk sac would be here. Amniotic cavity would be up here. Remember these layers? So there's an inner one and an outer one. You remember them? The one closest to the center is the splanchnic lateral plate mesoderm, and then there's somatic lateral plate mesoderm on the outside. So they're going to get similar names, as we'll see here in a second. So yeah, so it rips through and creates 2 new layers, and here we go. So same kind of story. So we just said this is splanchnic extraembryonic mesoderm. We know that already. So this is going to be called splanchnic lateral plate mesoderm. That's easy enough. And this one's going to be called, it's on the outside where the sun is, somatic lateral plate mesoderm. Now again, the AKAs will be coming at you, which drives everyone insane. So I have a whole slide on AKAs, but oftentimes this is just called splanchnic mesoderm. Splanchnic mesoderm. And this one's called somatic mesoderm. So when you hear splanchnic or somatic mesoderm, you think of these 2 layers of tissue. You do not think out here. They're not going to talk a whole lot about these up here. And they'd have to call this splanchnic extraembryonic mesoderm, especially at this point, because now we have an intraembryonic mesoderm as well. So make sure you know your anatomy here. Good. And then there's our intraembryonic coelom there, communicating with the extraembryonic coelom, aka chorionic cavity. All right, who cares about why do we need to know all this stuff? It just seems like ridiculous, doesn't it? Well, there's 3 really important cavities in the body that will come from this intraembryonic coelom. Who are they? They're the 3 P cavities we've talked about. I know I've talked about that before. 3 P cavities. So we have pericardial cavity, we have the pleural cavity, and we have the peritoneal cavity or peritoneal cavity, however you want to say that. That's the pericardial surrounds the heart. It's a little space between 2 serous membranes, if you will. Pleural cavity surrounds the lungs. Pleural cavity surrounds all the intestines and all the stuff in the abdominal cavity. Now let's bring those back to the top. Since this is such high yield board stuff, I think I can stick this on here and test you on this again. And I don't want to hear complaints like last quarter. Complaining. Oh, we've had this before. Why do we need this again? Because this is going to be on boards and it's coming and it's related to embryology. That's why you need to use this. So here we go. So this is sac around the heart. You'll remember it's called the pericardium. These should be easy questions, right? You've had them before. It's called the pericardium or pericardial sac. It has 3 layers. It has a layer on the outside that's thick and fibrous, so it's called the fibrous layer of the pericardium, fibrous pericardium. And then we have a layer that's a slimy layer that's stuck to the fibrous pericardium. And you can't separate them. You'd have to look at them with a microscope or electron microscope to see the difference. Those 2 layers are stuck together. And this is the parents on the outside. This is the parietal serous pericardium. And then we have another layer that's stuck to the outside of the heart. And that's called the visceral serous pericardium, aka epicardium. And that one's going to be on boards, I'm telling you, because that layer that you can kind of see, I kind of drew that in, photoshopped that in, that little grainy layer there, that is the outer part of the heart wall, and it's the inner part of the pericardial sac or pericardium. So it serves double duty. So pericardial sac is made up of an outer layer, fibrous pericardium, we said that. Everything I said right here. And then there's a space in between the parietal and visceral serous pericardia. And that space is the parietal pericardium. That space is the pericardial cavity, or pericardial chamber, or pericardial space, pericardial cavity. I can't think of any other words off the top of my head, but it's a space here, and it's filled with a little bit of fluid, but maybe I think it was 10 cc's of serous fluid, and that prevents friction buildup between the pericardium and the heart. Pericardium is a protective sac around the heart. Viruses get pneumonia, bugs try to invade. It can't invade very well. You can't get a pericarditis when you have pneumonia because of this pericardium. Okay, enough said about that. Same theory here with the pleural cavity. So the pleural is the same theory. It's a sac around the lungs, and it's a double-walled sac. And if we look at this piece of it right here and blow it up, we have the visceral pleura stuck to the lung tissue, and there's the parent on the outside, the parietal pleura. And the space in the middle is the pleural cavity or pleural space. And it also has maybe 10 cc's of serous fluid to reduce friction when the lung is constantly expanding and contracting, expanding and contraction. Okay, these are mesothelial tissues, not mesenchymal tissues. These really are mesenthelial tissues which secrete serous fluid. Peritoneal cavity, it's a little bit tougher to grasp, but there's 2 layers of peritoneum, and there's the parietal layer on the outside stuck to the like the abdominal wall here, the transversus, let's see, what would that be? Transversus abdominis stuck to the inside of that. And then the visceral peritoneum is actually wrapped around all these organs. So you can see it wrapped around the intestines here. It's wrapped around most of the liver, but not all of the liver. So here's a good place to look at it. So this is parietal peritoneum. This would be visceral peritoneum right here in blue, right? And the peritoneal cavity or peritoneal space, I mean, it looks like the other spaces here, but here it's huge. You got a lot of space. And so again, there's probably about 50 cc's of serous fluid that's normally produced, and it helps all the intestines glide and slide over one another. And you get elongations of the visceral peritoneum. And that's what forms the omentum. So the greater omentum is actually 2 layers of visceral peritoneum. See how that works? It's not parietal peritoneum now. Okay, so peritoneal cavity. See how that works? I know we went over that in SysPhys. I think I did at least. But if not, there it is. So what about pathologies involved with these cavities? Got to know these. Super high yield board stuff. So if you get a buildup of serous fluid from an infection, the cells get upset and they oversecrete fluid. Or you get an injury and blood fills those spaces, or pus from a wicked infection, or air from a puncture wound. You get what's called an atelectasis. Atelectasis. Okay, and that atelectasis is specifically for the pleural cavity. It crushes the lungs in atelectasis, and there's different types of them. And then if you get that same story, fluid, blood plus air squeezing in that tiny pericardial cavity, then you get not atelectasis, but you get a different word, cardiac tamponade. It's the same thing, exactly the same thing, just a different word. It's an overstuffing of that tiny little space with too much fluid, and it can crush the heart. It can kill you. And then the same story, if you get a buildup of fluid, blood, pus, and air in the peritoneal cavity, then we call that ascites. Ascites. Atelectasis, so really important you remember this. We have different types of atelectasis. The classic one is number 2 here, a compression atelectasis, where you get fluid, blood, pus, air, whatever, in the pleural space between the parietal and visceral pleural, and it grows, it accumulates, and it pushes or compresses the lungs. But there's other types of compression of the lung. You can have a reabsorption atelectasis, or sometimes it's called a reabsorptive atelectasis, and that happens when one of these big segmental bronchi get compressed because of let's say a blood clot. That would be on the outside. That's a bad example. Let's say the classic example is wicked severe mucus in someone who has asthma or chronic bronchitis. They make a mucus that is mucin rich and water poor, and you get a big old plug of it, and pretty soon you can't get air down into this part of the lung anymore. And all the air gets sucked out by the blood passing by, and there's nothing to replenish the air. And the carbon dioxide that was in there gets reabsorbed through the walls back into the body, and therefore the tubes collapse. And as they collapse the lung tissue with them. That's our absorptive atelectasis. Contraction atelectasis is some type of inflammatory disease that affects the lung stroma and probably the parenchyma as well, but it causes scar tissue, and the scar tissue contracts and starts to pull the lung tissue in. So emphysema would be a form of this, could cause this. There's a bunch of other ones that could cause it. Okay, here's an example of tamponade. So there's not different types of this, but in this case it looks like the heart got infected and the pericardium overproduced serous fluid, and now that fluid is building up around the heart and it's smashing the heart in. And so your ejection fraction or your cardiac output would be decreased because the heart can't, the ventricles are pinched and you can't fill these ventricles up with blood. So you get decreased ejection fraction. That's going to cause hypotension. Here's just an example of ascites. You can see the belly's fluid up. Alcoholics can have this, probably one of the most common causes of it in the Western world. And how do you tell this apart from a Homer Simpson kind of a fat-infested belly? It can be difficult, but there's a test called the fluid wave test that you can do. I'm sure they've taught you that by now. All right, just kind of a past slide, just showing you that extraembryonic coelom. Could have probably taken this out. But again, we had splanchnic mesoderm. They really would have to call that splanchnic extraembryonic mesoderm. And then we have somatic extraembryonic mesoderm. Okay, the two layers, we've talked about them. Let's look at them a little more closely. So the AKA somatic lateral plate mesoderm, also known as parietal lateral plate mesoderm, or any combination of these words, parietal somatic mesoderm, who knows what they're going to come up with, but the board books say either parietal lateral plate mesoderm or somatic lateral plate mesoderm. This one, you should know the anatomy. Where is it next to? So, somatic, sun on the outside, it's next to this. What's this layer? That's ectoderm. So it's good to know it's next to the ectoderm, because the combination of these two is going to create something. A new kind of a one word for two layers type thing. Splanchnic or visceral lateral plate mesoderm is next to the endoderm. Let's see if that's true. There's the splanchnic, always means toward the inside. Yeah, and there's the endoderm right there. And again, this author put this big space in here just to help us, but these would be stuck together in real life. Okay. Notice who's it continuous with? Splanchnic lateral plate mesoderm. It's continuous with somatic splanchnic mesoderm. Let's see if that's true. So splanchnic lateral plate mesoderm, and it's better to say it's continuous with this tissue, which was splanchnic extraembryonic mesoderm. That's who it's continuous with. Is that what that said? Yeah, that would be pretty tough to say that. So if I asked a question like that, I would call this, just to make sure I keep it as easy as possible. This is splanchnic extraembryonic mesoderm. Or this is splanchnic extraembryonic mesoderm. That's endoderm here. All right. Oh my God, the AKAs will drive you insane. Again, somatic lateral plate mesoderm, somatic mesoderm, sometimes they just call it. Parietal mesoderm, parietal lateral plate mesoderm, somatic intraembryonic mesoderm, somatic pluric mesoderm. Oh my God, drives you crazy. Same with splanchnic lateral plate mesoderm. Okay, parietal mesoderm. And Morse uses this visceral mesoderm. So that's kind of confusing. I would really, all the other authors use splanchnic lateral plate mesoderm, but Morse doesn't. And that's a chiropractic board book, so I worry about visceral mesoderm. You should know that AKA. That's an AK for splanchnic lateral plate mesoderm. And I get it because that visceral layer of all of those 3 P cavities comes from this. And that's why he uses it. Same up here. For somatic lateral plate mesoderm, he uses parietal mesoderm. And I get why he does that, but no one else does it, and it gets confusing for the student, I think. So it'd be nice if he just followed in line with everybody else. Just another picture. Somatic lateral plate mesoderm, splanchnic lateral plate mesoderm, splanchnic extraembryonic mesoderm. Now we have this labeled correctly. Somatic extraembryonic mesoderm is right here. Great. Always confusing. That's the concept of the splanchnoplur and the somatoplur. It's really not that difficult. Let's just look at the picture. So basically, if you combine the somatic lateral plate mesoderm with the ectoderm, which is happening in real life, there's not a space here like this author's showing. If you combine these two together, then we can say one word. We say somatic pluric mesoderm, or sometimes just somatoplur. So somatoplur, make note cards. Somatoplur, what's the AKA for somatoplur? Somatic pluric mesoderm. What does it consist of? It consists of ectoderm, and it consists of somatic lateral plate mesoderm, is what the somatoplur consists of. Then we have the splanchnopleur, is these two layers squished together. Okay, and again, there's no space in between there. So splanchnopleur, aka splanchnopleurotic mesoderm. What makes up the splanchnopleur? Well, it's these two layers. So we have endoderm, and we have this layer. What was that layer? You can look over here, splanchnic lateral plate mesoderm. So guarantee there's questions on this, so make sure you understand that. Everything I just said is right there. I think that is enough for your brains. That is the famous Lola who grew up with a red-tailed hawk, thinking that the red-tailed hawk was her sibling and treating her very well. And that story, I have a YouTube video on that whole story, kind of sad ending, but this is one of her first kills. I was on vacation, so I spent every day up there watching this bird grow up, and this is the first kill that she ever got, a squirrel, and she was bringing it back to the nest to eat it. So very exciting time for that story. All right, email me questions, and if all goes well, I will be back on Monday or certainly on Tuesday. All right, see you later.